This production was recorded at the podcast studios in the Media Arts Centre at Murdoch University. The views and opinions expressed in this podcast are solely those of the hosts and guests and do not necessarily reflect the official policy or position of Murdoch University. Any content provided by the hosts and guests are their personal opinions and are not intended to represent the views or opinions of the university, its administration, faculty, or staff. Now we have that out of the way, let's talk science. Okay, well, today's guest, I have Shannon Dee, who's a close friend of mine, and we have been working on a project together over the last few years with a few field trips up north, which is not what we're going to be talking about today, but Shannon is a very high-achieving woman that works across quite a few different areas, with her expertise, though, is in corals. So today we're going to be talking about corals, and for the first question, first I'm going to say thank you for joining us.
No worries. Thanks for having me.
This will be going to be interesting. It's interesting having like a friend joining on me interviewing you. I've obviously spoken to you for a long period of times, many times before. So there's sure there's going to be a lot of inside jokes or something that maybe come across, but
A bit professional.
Yeah, hopefully. Well, tell me, what is a coral and what is a coral reef?
Okay, I feel sorry for anyone who's not watching this though, because I'm going to use my hands a bit.
It's okay.
So a coral, I'll just focus on hard corals. So a coral as we see it is actually a colony of individual little animals, which are the polyp. So a coral polyp basically looks like an upside down jellyfish. So it has like a central mouth and a bunch of tentacles around it. So different types of corals have different forms of polyps, different sizes of polyps. And within each coral colony, which is that structure that you see, each of those polyps is a clone of each other. And then so a coral reef is built up of different corals all building together with their calcium carbonate skeletons along with other calcifying organisms and taxa that then build up that solid reef structure.
Right. So when you're saying they're a clone of each other, does that mean they are producing asexually or are they?
So like as the coral grows, I'm not actually like 100% sure, but like as the coral structure grows, the polyps just kind of like regenerate and bud off from each other.
Oh,
right.
Okay.
So it's not like.
So is it one big living organism? Or is it lots of little bits?
Lots of little bits that share the same skeleton.
Okay. So
each individual polyp eats, like feeds by itself, collects its own energy and reproduces.
Right. So that's not the zooxanthellae that's inside. This is because that's another organism, right?
Yeah, that's a
symbiotic relationship.
Symbiotic algae, which coral hosts.
Right. How does that work?
So basically, corals themselves or the polyps don't really have complete digestive systems, or like they kind of. I'm just going to rewind. So the corals take on this zooxanthellae algae to help them basically gain energy more efficiently. So for an algae's point of view, the coral polyp is a safe place to live and it's also a convenient location where it can photosynthesize. And there's other chemistry points, which I'm forgetting off the top of my mind. Mm-hmm. And then in turn, the coral polyp gets the sugars and proteins from the photosynthesis, which then it uses for energy to produce its skeleton.
Right. How fast does a coral grow?
Depends on the coral and depends on the environmental conditions. So like massives or encrusting corals tend to grow slower. So that could be, 1 to maybe 6 millimeters a year.
Whoa.
And then like staghorn coral, the big branching finger coral, I think can go like 13 millimeters a year or more, like in extension rates from the fingers.
So those big events where you lose mass coral, that's thousands of years of growth.
Those big bouldering corals, the ones that you see like the size of this room, that is thousands of years old.
The size of this room is probably about 3 by 3.
Yeah, and like another 3 meters tall. Those big bommies are really, really, really old.
Right. So it's a real shame when that happens, which we'll go into a little bit later.
Yeah.
But I want to go into what is a turbid reef, and then we're going to talk about your paper, which was part of your PhD. And that, because it was a super fascinating thing. So your paper is on the application of carbonate and sediment budgets to assess the stability of marginal reef systems, which looked at the effects of anthropogenic impacts like climate change and sea level rise on coral reefs and the associated landforms.
Yeah, that was the big kahuna of my thesis.
Yeah. So let's elaborate on that. Let's go with that. So let's start with like, what is the fringing or turbid reefs in these areas? What coral type of reefs are we looking at?
So for my PhD research and like an area that I'm quite passionate about is looking at turbid reefs and their forms and functions. So most of my research has been done in Exmouth Gulf, which is a naturally turbid area, but it's not a constant chronic turbid area. It's a fluctuating there's fluctuating turbidity along that coastline. And so by naturally turbid, I mean I'll go back to actually what is the turbid water. So it's basically like the water column has lots of suspended particulate matter or particulate organic matter in it. So it's not like a clear water where you have tens of meters of visibility. Sometimes you can see 30 centimeters.
Make it hard for those photosynthesizing zooxanthellae, right? Or
yes. So so corals in these kind of habitats are seen usually, like classically, everyone would expect there to be low coral cover, if any at all. And the corals that are there are expected to be unhealthy and struggling. But now researchers over the last 10 years have been finding coral reefs living in these habitats and thriving and having some growth rates comparable to clear water, beautiful tropical reefs. And these conditions are expected to increase with climate change, but then also direct human influences. So I said, like, you can have naturally turbid reefs, but you can also have human influenced turbid reefs. So areas where there's lots of dredging or port development or coastal urbanization, lots of pollution from river runoff from agriculture and stuff like that can cause a lot of turbidity.
Are these species of corals that are in these reefs, are they different species, or are they just sort of, I guess, genes of the standard species we see that have adapted to live in these environments? Or
so there's I'm forgetting what it's called, but the triangle of life history stages or whatever it is. So it's originally from plant biology, but it's been adapted to look at corals. Emily Darling did a paper in like 2012, which goes into it really well. She listed 4 life history stages, but I'll just focus on the main 3. I think it's and now I'm going to forget them as well. So there's like the competitive weedy type. So they come in after an event and they thrive, kind of thing. They take over.
Oh, the pioneering species.
Yeah. Then there's the
secondary colonizers. Is it these?
No, like the 3. There's There's ones that do just well when it's like nice and healthy and everything's good. There's the competitive weedies.
Opportunistic?
No. And then there's the like stress tolerant ones. So turbid reefs are usually
it's the end of a big work day. So don't worry. It's completely I know what this mental blank is like.
Yeah. So anyway, those are the three kind of. So on your beautiful Clearwater reefs, you usually have those ones that go well, the top triangle ones that I forget the name of. And that's a lot of your acropora branching, like your big blue staghorn corals and stuff like that. And then in your turbid reefs and other more marginal reefs, so like mesophotic or cold water or, not optimal condition reefs, you have more stress tolerant species. So that's more of your like massives and submassives and encrusting corals. And also turbinaria, we have a lot of it in WA. It's one off of Esperance.
The big plate coral.
Yeah, it's like a foliose. It can be like a plating or like a little vase type looking thing.
Looks like a rose. Like
kind of, yeah.
Rose, but as a coral.
Yeah.
Yeah.
So yeah, those are kind of the more species that you have on turbid reefs.
Mm-hmm.
And I forget if you asked something else.
Actually, I can't quite remember my question either. But so how do you measure their like success, or like what is, you look in your paper, it literally talks about carbonate and sediment budgets. What is a carbonate and sediment budget?
So a calcium carbonate budget is a good kind of assessment for looking at more of a holistic view of a reef's health. So basically it's looking at the inputs and outputs of calcium carbonate from a reef system to assess the net growth of the reef, the net accretion.
Mm-hmm.
So the main inputs are coral growth. So you do that by assessing what corals you have there. You can do direct measurements of coral growth rates, which is highly encouraged because they can vary so much between reefs.
So when you're measuring a coral growth rate, are you like going from like the nucleus of the coral, like the centerpiece, and then measuring outwards? Or like how do you like know, especially because like 1 millimeter, if it's in a year, because your PhD obviously is you do like 2 years of field work or so. That's, you'd be looking at like 2 millimeters of growth.
Yeah, well, you do. People have always done it. Well, not always, but so corals have growth bands.
Mm-hmm.
So a lot of people, especially with the acropora, you can break off the fingers and like cut it with a rock saw and look at like measure the growth bands.
Oh, same as like tree rings or when we're aging fish, looking at the otoliths.
Yeah. And then, but yeah, we also look at calcification rates and the net weight of the corals. So for my study, I used the buoyant weight method. So basically we like collected a whole bunch of little nubbin, little sections of the coral colony. We break them off and take them back to the house and attach them to little plates. And so we would weigh the coral by itself, the coral with the plate. And so that's your like starting weight.
Mm-hmm.
And you're weighing it in water as well, so it's buoyant. And then we put them out on a big tripod on the reef and left them for a year or so. And then we came back and collected them all and weighed them again to see how much mass they had accumulated over that year. Yeah.
Wait, so when you said you put them back out on a tray, that was back in the fringing reef, back on the
turbid reefs,
back on the turbid reefs. Okay, so they were still living; they weren't dried out.
No. Like when you break them off, you put them straight into water, and then overnight.
Kind of like how they do coral restoration, like tidal.
Yeah, coral gardening.
Coral gardening.
basically, yeah.
Yeah. Okay. Cool.
So it was like that, but yeah, and we had them in like little milk crates in the canals in Exmouth overnight.
Yeah.
So to keep them happy.
Yeah. I can imagine it being a pretty fun project.
Yeah, long nights.
How do they accrete carbon? Like obviously, I guess in the same way as like
calcium carbonate.
Yeah, calcium carbonate. Why? Like, how is that happening? Like, why is it like impacted by temperatures? Like, is it faster or slower in warmer or colder temperatures? Like, ocean acidification, I'm assuming would affect it as well. Like, what are these variables that you look at that could be affecting these corals?
Well, yeah, I mean, every environmental factor,
Mhm.
it's all cumulative impacts onto the coral. So, and in this case as well, that it's that zooxanthellae, the symbiont, that is producing the energy that the coral needs to make its skeleton, so make the calcium carbonate.
So in the reefs you were measuring, was there like certain reefs that were better? Like where they grew faster, like on certain islands or were they all sort of growing the same rate?
On some of the less turbid sites, I think we got like slightly higher growth rates, but one of our more turbid site at Fly, it's quite close to the Ashburton River mouth. So it experiences high amounts of turbidity when the river's flowing. So and then with that as well is more sedimentation as well, which the corals need to express more energy to basically clear out their little polyps when the sediment lands on the polyp. They need to use energy to
expel it.
Yeah.
Yeah.
So instead of, putting all of their energy into growing their skeleton, they're also doing like maintenance cleaning and stuff.
Right. The zooxanthellae don't help out with that?
Well, they give them the energy to do that.
Okay.
Yeah. But then also, as you kind of mentioned before, but we didn't really go into, with the shading from that from the turbidity, the corals can't always depend on the symbiont to photosynthesize and give them the energy that they need. So corals in mesophotic areas have been shown to have adaptive trophic or feeding strategies. So they'll instead of just being an autotrophic feeder, which is through photosynthesis, they can be mixotrophic and use heterotrophy, which means they actively collect their own food. So they'll use their little tentacles to grab particulates from the water column and get energy and food source that way as well.
Right. Well, in terms of like the types of energy, can you measure like which way they're receiving their energy from? Like, is it mainly from photosynthesis or is it mainly from these other ways? Like, is there ways to prove where they're getting the energy from?
Yeah, so you can do that through stable isotopes. So you can use bulk stable isotope to basically see what level of reliance the coral has on autotrophy photosynthesis or heterotrophic feeding. So if it has a higher carbon percentage in your stable isotope results, a bit jargony, but the coral will be more reliant on autotrophic feeding, and if they have a higher nitrogen ratio, then they're more likely to be feeding heterotrophically. And I'm doing a study at the moment on the side of the project that we're working on the intertidal areas. I've sampled a few of those turbid reefs right at the creek mouths in the same turbid area to look at compound-specific stable isotopes. So that kind of goes into just like a little bit more detail. It looks at, for each of those coral samples, we split the coral host tissue, so the actual coral polyp tissue, and the symbiont algae. And then for each of those, we basically extract the amino acids that each of them have and then run the stable isotope carbon nitrogen ratio on each of those amino acids. So then that gives each of those, like the coral host or the symbiont algae, as well as source material that we're testing as well, like mangrove leaves and cyanobacteria mat and POM. It gives it a carbon signature. So each of those source materials will have a signature of amino acids at various levels. And then, we're picking that up in the coral. We can tell if the coral is specifically dependent on that food source. And then through this as well, like splitting the host and the symbiont, we can see how reliant the actual coral host is on that symbiont as well.
Is there any results from this? Did you do that in your PhD as well?
No, I've done that this. No. I was lucky enough for ECU to give me some additional funding.
Okay.
To go and have some fun. I sent samples off. Mm-hmm. I collected them last year and did the preparation at the end of last year and sent them off just before Christmas. So hopefully I get some results soon.
Without having any data on that, what do you think what's their main source of energy? What are you predicting?
I am predicting them to be at least somewhat reliant on heterotrophic feeding.
So that means they're getting their nutrients from the outflow of these creeks?
Yeah, so I collected coral samples on the end of the falling tide when it was really turbid. And all of the corals had their little tentacles out. It was really cool. And it was like super murky, but when you went up close to the coral, you could see them with their little tentacles out at each of the polyps, just like taking in little bits of particulates. But we looked at different species of coral, so like favids with big polyps to parietes with tiny polyps and acroporas with different structured polyps. So we'll see what different.
Right. So is this going to be feeding on like it's organic matter, right? Does that mean it's the algae coming through that, or is it fish poo, or like just what's the matter that they're feeding on?
The analysis is quite expensive.
Mm-hmm.
So I'm only looking at cyanobacteria mat, which is a massive potential source in the Pilbara. Mm-hmm. Mangrove detritus, so leaf litter.
Where does cyanobacteria mat grow? Sorry.
Where?
Yeah.
Everywhere.
But the main bits are it's more the back of the creeks.
Oh, yeah, sorry. Yeah. In these systems, it's behind, so these are intertidal flats basically that run along the whole majority of the Pilbara coastline. So at the front you have your big beautiful mangroves, and then little creek systems go up between them, and behind your mangroves you get these kind of like mud flats, which then once they become flooded with water on the big tides, it all evaporates because it's so hot and sunny up there, and that leaves salt behind. So people either call them like mud flats or salt pans, salt flats. But in these areas as well, there's these massive cyanobacteria or microbial mats that thrive in the hot and salty environment. And other arid coastal areas around the world, it's quite common as well. But I mean, I'm finding they're growing everywhere.
Yeah. Well, algae is a very successful type of plant.
It's a microbe.
The
microbe, cyanobacteria, but different species of cyanobacteria.
Yeah.
But anyway, back to the original thing. So as the tides come up, we've seen that in these cyanobacteria mats, the water that's pooling on them is really high in nutrients. So we're expecting some of that particulate matter and the nutrients from the cyanobacteria mats as it's flowing out could potentially be a major source of food for either corals or other organisms in the food web in the Gulf. So
Yeah. So this is when you're talking about how far this extends to. Is this the whole Gulf, or is this like 20 meters from shore? What's like the length of like a turbid reef, or the bits that at least you're measuring in this area? Because I know some of these reefs that are there. This is not inside the Gulf. This is like if you go straight out, it's just straight ocean. So how far out are you thinking?
We're referring to Exmouth Gulf, by the way.
Yes. Yeah.
So turbid reefs are usually near shore, so within 10 to 20 kilometres offshore. I think it's more like 10 kilometres. And they're usually between 10 and 20 metres deep, so they're quite shallow.
That's
quite
actually
near shore.
a lot further than I was thinking. Like in my mind, I was actually only thinking like 2 meters deep, like 100 meters offshore. Like these are like the main, well, I don't know, recreational spearer, but like a lot of the main spearing grounds. So this is going out a long way from these areas that you're talking about.
Yeah, and like I said, the Pilbara is quite fluctuating turbidity. So on big rainfall events like cyclones, that turbidity plume, gets to that full extent. But other areas like on the Great Barrier Reef, there are areas on the inner Great Barrier Reef is, 10 kilometres or more ashore. I think Low Isles is like 7 nautical miles.
I
can't remember the convention. But yeah, so these inshore reefs like Palmer Shoals and somewhere else, I forget the main one. My PhD supervisor would be shaking her head. Those reefs are kind of more chronically turbid all year round and a bit like further offshore than what we're seeing in the Pilbara.
Yeah. I interrupted you before when you're saying the other things you're measuring. So it was a cyanobacteria mat, mangroves, like the part like
the detritus, the fallen leaves that are then decaying.
Mm-hmm.
And also cestin, so particular organic matter in the water column. So we just collected that by towing a net behind a boat for a few transects at the mouth of each of these creeks at the end of the falling tide.
You said it's quite expensive to analyze these samples.
Yes.
Is there a dollar figure? Is it like kind of standard across, like for your PhD when you're doing this? Is it the same price what you were doing, or is it like wherever you send it's always going to be a different price? Like what is this rough dollar value?
Well, the compound specific stable isotopes are still relatively new. They're not done as broadly as bulk stable isotope analysis.
Okay.
Like I struggled to find anywhere in Australia that would do it. I looked at a few places in America that were going to be about $400 per sample.
Whoa. So, and just so for context, how many samples, because it's obviously got to be replicates, and then if you,
I literally sampled like the minimum amount of stuff. That's why I only had 3 sources. So I had 150 samples all up.
Whoa.
Yeah. So
that's a lot of.
This is why I stress about any little bit of money being spent on field trips. Analysis costs a lot of money.
Yeah.
But yes,
don't need to go into my eating habits on field trips.
But no, luckily, yeah. So, and as well, a lot of facilities, they have the equipment to do the work, but it's really tedious and takes a long time. So that's kind of where most of the dollar value comes from. So a lot of facilities are happy for you to do it if you go there yourself and do it. But then you've also got to take into account your time and
flights, because
accommodation.
Obviously a new country. From this project we're talking about, they got sent to Hong Kong, right? Or is this the?
Yes, so we're sending our samples to Hong Kong University because they've been really great and they're more affordable, closer to home as well. And colleagues in Singapore have used them before and given good reviews. So
okay,
fingers crossed.
Yeah, fingers crossed. What can cause a reef to erode? Like, did you find this at all in any of your research? Like coral reefs eroding?
Yeah.
Yeah.
So, so coral reefs can erode from a number of ways. So physical erosion from cyclones or big storms, um, chemical erosion from chemical dissolution, so which increases with ocean acidification, so breaking down the aragonite.
Hang on.
In the calcium skeleton.
Elaborate, because I don't even know what aragonite is.
It's an element.
Okay.
I am not the best at the chemistry side of these things. So
yeah,
dissolution
for a future time.
Yeah. When I'm fresher in the morning after a coffee, maybe. And then the main factor of erosion really is bioerosion. So that can be from your big external eroders like your parrotfish and urchins. And then you also have endolithic erosion, which means like internal erosion. And that's from macro and micro borers. So macro borers are things that are easier to see. So your bivalves, worms, and sponges.
Most of them are natural, but there is non-natural ones, right? That have been like introduced species kind of. Is there?
Yeah, I'd imagine so.
Yeah.
But like it's all natural. So usually when a bit of coral dies, and it doesn't have that protective flesh on the outside, it's first inhabited by microborers, so like fungi and algae that kind of weaken the external skeleton, and then that allows those macroborers to kind of as succession come in and break through and bore into the skeleton. And then when the skeleton's full of delicious little algae and worms, that's when the parrotfish love to come in and just chunk at it.
Oh, so they're not eating the coral; they're eating they get the things inside it.
Well, yeah, indirectly they eat the coral skeleton, which then
they chomp it up and
then they poop it out, and that makes sand.
Yes.
Yeah.
Yeah.
So going back to your first question, ages ago, like when you asked me about a sediment budget.
Yes. Yeah.
So one of the main inputs of a sediment budget is bioerosion from animals.
Right. So in a way, I guess like parrotfish do harm by eating corals and taking away all this years and years of growth.
It's a balance.
Yeah, but then
their poo helps sustain, because that sediment as well that's produced on a reef system, not only from bioerosion but breakdown of like physical erosion and stuff, as well as sediment getting transported onto the reef. That sediment can then stay within the reef system, like filling little gaps and stuff. To help solidify the reef system, or it goes towards adjacent landforms, like if it's a reef island, like in my case, or coastlines if it's a fringing reef, or it can get transported offshore and we don't worry about it.
Bit of a love-hate relationship with parrotfish then.
Yeah, so that's a good, that's the thing about turbid reefs. Parrotfish don't really like turbid water. So the bioerosion rates on turbid reefs tend to be a lot lower because you don't have the big chomping parrotfish.
And the sediment rates would be a lot lower too, I'd imagine then. Well, I guess then you got longshore drift and like all the oceanography.
Yeah, there's a whole lot of hydrodynamics.
Yeah.
Things to go into it. I didn't do a very from that full extent. There are some very, very detailed coastal engineering sediment budgets, which I just did a basic kind of ecology sediment budget in my PhD compared to those really complex.
So when you say like what a budget, it's the budget to me in a way, I know we sort of touched on it earlier, like I keep thinking of like, oh, I got to budget my money so if I want to afford this or whatever. How does that relate in coral like budgets? Like what, I know we've said what is a.
So you have your income, which is all the things making calcium carbonate on your reef. So as well, we also cut this off before.
Yeah.
So you got your coral growth.
I just needed to be explained again to get it through My head.
You got the coral growth, but then you also have other encrusting organisms such as like Christos coralline algae, like CCA. Also like different types of worms make little calcium carbonate encasings and stuff. So that all builds up to that they're all your inputs. And then your outputs are those different forms of erosion. And then that net accretion, or, it could be negative accretion, is your result of your budget. So it looks, what have you got left at the end of the year? Are you going to accrete by?
So is this then used in like to model?
Yeah. So then it's used to, so people use carbonate budgets basically to give a rapid assessment of, that full accretion rate of your reef system. And then you can potentially, my PhD supervisor, Nikki Brown, is looking really in depth at making a full model of all of these different inputs of environmental conditions and bioerosion conditions and XYZ that are going to bring in and all impact that final number that you get. And so then we can look at under different climate scenarios and different, if there's a cyclone, model those potential
Well, I guess
impacts.
the main one at the moment is coral bleaching.
In WA, yeah.
Yeah.
Well, globally really as well.
Yeah. What is, I know this is completely off what we just talked about, but what is coral bleaching? Like, how does that happen?
So when there is something that's quite stressful to a coral, such as thermal stress, if a coral is exposed to abnormally high temperatures for an extended period of time, it can be quite stressful. But then when you also accumulate that with intense radiation, so if it's middle of summer, it's very, very bright, or other stresses such as like high amounts of nutrients that could be toxic to corals, or if there's an anoxic episode as well. In some cases, turbidity as well. So it's that accumulation of usually it's the main driver is higher temperatures. So yeah, basically the coral gets stressed and it wants to detox. So it expels that symbiotic algae.
The zooxanthine, because that's what gives us its colour.
Yes.
Yeah.
So through the photosynthesis of the zooxanthellae, that's what exposes the colour on the coral. And then, once the coral gets stressed out and sheds its algae, what's left is the translucent tissue of the coral, which then shows its white skeleton. So in this state, the coral isn't dead. It's just
under a lot of stress.
It's very, very stressed, and it's not getting the energy that it needs. It's not taking care of itself. So if these conditions continue for a couple of months, then the coral effectively kind of starves to death. But if conditions go back to being okay for the coral, then it can take on that algae again and get back to its normal health.
From your research, would that help help policymakers or other managers helping them in helping to preserve or conserve these corals, understanding the budgets, the coral budgets and sedimentation, that would because would be an impact that would influence those budgets, I would imagine.
Yeah, so we've seen over the past 20 years people apply carbonate budgets before and after massive heatwave bleaching events to see declines in that ultimate production of the reef. And also after coral bleaching events, we've had massive, I think in the Caribbean, after a big bleaching event, there was a massive urchin feeding or something that, so after these events, the reefs can just become decimated. and these carbonate budget tools, especially with Reef Budget that's come out of the University of Exeter. No, yeah, something like
Yeah. I think that's it.
Yeah,
Sounds very Harry Potter-like, actually.
yeah, Chris Perry and Innes Lang, their group have made the Reef Budget page on the. They have a web page where you can basically go and get these interactive spreadsheets. So you don't need expensive equipment or anything. You just need to get out onto the reef, and it gives you all the parameters that you need to measure, and you put them in there, and you get an instant number. So it's really, really accessible to do these assessments for people. It doesn't matter, you could be at a remote atoll in Indonesia or anywhere, and as long as you've got internet access, you can do a rapid assessment of kind of what state, positive or negative, your reef structure is in.
As in, like, community people can do this? Or?
Yeah.
Yeah, right. So they can, and then is there a platform that they can release, like give this information to scientists, or like?
No, there isn't, but there really should be.
Yeah, because it's kind of like, so they can do it.
Yeah.
And then they just know. Is that what it?
Well, it's mostly for scientists at the moment.
Yeah.
So, but it's giving that opportunity to scientists in developing countries, coastal countries as well, and especially those countries that are based on low-lying islands, who kind of need to know, are these reefs going to continue to accrete, or are they struggling, because then those reefs are what is breaking down wave energy as well, which helps protect those islands from inundation and erosion.
Surfers would not like that.
Hey, reef breaks are sick.
Yeah, but they want more energy, more waves, more surf.
Yeah.
But that's obviously well off the point. What future research directions do you think are most critical for understanding like these reef sustainability?
Well, generally marginal reefs. So these are reef systems that exist in non-optimal conditions. So turbid or mesophotic or cold water reefs are incredibly understudied because they're not fun to work in and they're not pretty.
Wait, are these talking about the reefs down on the south coast?
Yeah, they would be considered marginal reefs.
Yeah.
So, I mean, these reef systems host corals and other benthic taxa that, are withstanding these conditions. And, there's been so little work done onto looking at their physiology and how they have adapted or how they're existing under these conditions. Like the corals in Exmouth Gulf, they experience temperature fluctuations from 33 degrees in summer to 18 degrees in winter. That's like 15-degree fluctuations, which is insane for a tropical reef. And then the salinity's around mid-40s in the Gulf. Yet hardly anyone has looked at the physiological adaptations of these corals and how they differ from clearwater reefs on neighbouring Ningaloo.
So you wouldn't really class them as a stenohaline species. So for like in terms of marine fish, if you go 0.5 off a salinity, a lot of them die. Like in estuaries, they can withstand massive differences, but like marine fish, if you go like to 37, 35, well plus like that, but many of them just die straight off. And these corals are able to adapt or live through that, which is quite crazy. So that's what you're saying. People need to be sort of more researching these adaptations that these corals have for the future.
Yeah.
It's not saying the ocean's going to get saltier, but those other like heat stresses and things.
Yeah. And turbid reefs are expected to increase with sea level rise and continued urbanization of coastal areas. So, having an understanding of what's going on those reefs and how the corals are withstanding that, and then that can help inform us about how corals in other areas that are going to become turbid, or corals in our tropical reefs, which are so severely impacted by bleaching, how they could potentially adapt or how we can modify them to help them adapt.
Right. You mentioned sea level rise. How does that impact corals?
So, I mean, it's kind of like a catch-22. There's the one argument where if sea levels rise, it gives more accommodation space for corals to keep growing. And then the other side of the coin is if sea level rises, then the water's getting deeper, which then, makes it harder for corals to
Right.
photosynthesize. And then also if, in these turbid reefs or
But then
stress
couldn't they
reefs.
just adapt to heterotrophy
Yeah,
feeding?
yeah. Yeah, we don't really know because,
Mhm.
I mean, in the time span that people have been doing in-depth studies of coral, there hasn't been much sea level rise, and I'm not totally up to speed with the reef geomorphology.
Mhm.
There's another supervisor that's going to be sad
Yeah.
with me. Sorry, Mick. But yeah, I think.
Yeah.
Yeah.
Well, I did touch briefly at the start of the podcast that you've been quite high achieving, and you've worked in quite a few different areas, obviously mainly in the corals and stuff, but you've got a few other outside projects that you've been involved in, and one of them is you've been selected as one of 60 women worldwide for this Homeward Bound Leadership program, which aims to enhance and influence the impact of women in science. That is very impressive. You also
Women in STEM.
women in STEM. You also, is it president of AMSA?
I am president of the Western Australian branch of AMSA, yes.
Yeah.
Which is the Australian Marine Sciences Association.
Yeah. You seem to be a part of everything. And firstly, I want to go back to the Homeward Bound Leadership of Women in STEM. What do you do there? Like, what is this?
So this is a really incredible initiative. I'm in Homeward Bound cohort 9. So before me were 8 other cohorts. So basically Homeward Bound gets women from all over the world who are in science, technology, engineering, maths or medicine, and basically puts us through a year of online learning and interactive learning to develop leadership skills, and not just me being able to delegate to you what to do.
For this reference, Shannon is the postdoc on this project we're working on, so she's essentially my boss.
But I'm a cool boss.
Yes, very much.
But so basically the program really takes you at the start, it really gets you to reflect on what your values are and, why you're doing what you're doing. And basically, we're encouraged to kind of like map out what we want to be in different aspects of our life, but we've got to really reflect on those core values that we've identified in ourselves. And then take that and do a hypothetical map of where you want to get to in each of these areas, to kind of really encourage you to make sure you're making decisions based on what's true to yourself, so you're not just
what aligns with your core values.
Yeah, so you're not just like ticking boxes and blah, 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 but you're actually trying to bring authenticity.
Do you think there's a lot of that in science, just ticking boxes?
I definitely think like the classic academic.
But you've worked in consultancy and academic. I've never worked in consultancy.
Yeah, but I don't think it's just science and stuff. I think it's just everything in life, You're like, oh, I go to high school and then, I got to get certain grades and then I got to go to university because everyone's going to university. And like, I guess I'll get a job and like, I've got to earn a salary so I can buy a house and blah, 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 blah. And, you're just kind of like going through the phases of like having a career and 9 to 5 and then going home and blah. But, if you kind of actually look at it from the point of view, that whole thing of like, if you love what you do, you never work a day.
Yeah.
But kind of grounding that to the point where like you're trying to be mindful of each of the decisions that you make. You're not just going to say yes to a job because you get offered a job. It's say yes to the job that is actually going to be fulfilling, you know, and trying to actually strategically think about what avenues you want to do. Or like, it might encourage you to slightly change the trajectory of your career. But anyway, like, so this whole thing, it started off as, really aligning what is authentic to yourself. And then it goes into the chapter of, leading yourself. And so it's not just straight into blah, 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 but it's like a lot of the foundations.
Taking control of your own.
Yeah, a lot of the foundations of the program are, how to actually manage yourself. And and then we also get in contact with some leadership coaches who.
Is this program already started?
Yeah, I'm like halfway through.
Right. Well, that'll explain some of the emails.
I've matured so much.
Yeah. What advice would you give to young scientists then from what you've learned so far?
Well, yeah, just be authentic. I mean, I've kind of always been I always wanted to be a mermaid when I was little. So I was always going to do marine science. So I've kind of been lucky that it's worked out so far.
Yeah, I've always wanted to be a fisherman.
Exactly. We're just chilling.
Yeah.
I think it's, find what's fulfilling because, I mean, even with the work that we do, I mean, it's so cool and we get to go to the coolest places ever. And, so many of our friends probably look at our Instagram feeds and be like, oh my God, I'm so jealous, blah, blah, blah.
I think some of them also go, stuff that's muddy. That's hot, stinky, muddy, lots of sandflies.
Yeah, that's the other side of the coin is like our work is so exhausting. And then also we spend so much time in front of computers doing analysis and stuff and then your code isn't running and then you submit a paper and you think it's the best thing ever and it gets rejected.
Or no one reads it.
Yeah. So
that's a whole nother.
So science can be really defeating if you let it be defeating, but it can also be incredibly inspiring and just incredible. I mean, if you just kind of try to see, there's so much extra work that scientists do that isn't, appreciated or acknowledged. Reviewing papers for journals.
Yeah, you don't get paid to do that.
No, but, everyone's always like, oh, this reviewer was such a butt because they didn't like what I'm
Yeah.
But it's like that's someone who's literally volunteered like hours of their life to try and help you make your paper better, So
yeah,
it's kind of
seeing it from the other side.
Yeah. So, I mean, I always say like doing a PhD doesn't make you smarter. It just makes you more resilient because it's just 3.5 years of like banging your head against a wall and nothing's working and no one's showing you appreciation.
Thanks for telling me that at the very start of my PhD.
Yeah. And then it's all just for like a piece of paper.
Yeah.
But in hindsight, it's awesome. I think science can be so incredible if you do take care of yourself and you do prioritize. And also it's so incredible. Well, it's so important to have a good network because.
What do you mean by a good network?
So friends in science. So, if you have, it's the people who are around you, it's your colleagues. So if you have a supervisor or someone, if your boss directly isn't mindful of your well-being, then it's going to be really exhausting and draining working for them. But then if you have a supervisor or boss who's like incredibly understanding and, understands that people need rest and comes to you and asks you how your weekend was before, asking like, where is XYZ, then you're going to have a better experience. And then also with your networks, the more people you meet through avenues like AMSA, the more friends you make with similar interests, but then also who are people who you can turn to for help with your work, and then also people who will want to collaborate with you, and it makes it more fun that you're like doing things that are interesting with friends, and then you go to conferences and there's more friends everywhere. And
I can only speak from the marine side of things, but it is a very sociable field of work.
well, yeah. So recently I was very lucky that AMSA sent me to Science Meets Parliament.
Oh, yes. Yeah,
so-
I know you're very high achieving. It's crazy the things that you have done.
It was so cool. So Science Meets Parliament is basically, I think it was like 500 scientists from around Australia. I think there were engineers as well. Basically get invited to go to Canberra.
Not just marine scientists.
No. So get invited to go to Canberra, and we have like a 1 day at a hotel in like a big function room.
Mm-hmm.
And it's seminars and workshops and stuff about, about like addressing scientists, addressing science.
In Parliament.
Yeah, or like communicating science to policymakers in ways that they will care about, and also kind of looking at from the side of the policymakers. And that's second day you get to actually go to Parliament House and meet a senator. And
Who did you meet?
I met Karen Grogan, who is a Labor senator from South Australia, and she was awesome. So we're friends on LinkedIn now. So shout out Karen, my girl. She was awesome. And so the cool thing about that, you say like marine scientists are very social and fun, but here we were with all different kinds of scientists and engineers and stuff, and everyone was so social. Literally, like you couldn't stand there and be awkward in the corner because someone would just come up to you and be like, "What do you do?" Like, wow. And everyone, and because, it's not just like, "Oh, I do fish," like, "Oh, I do fish too." It was like, "Oh my God, you're like a neurosurgeon."
Yeah.
So you're meeting people. Everyone's so exciting because everyone's
doing. Did you get some contacts for me to bring on the podcast?
Karen. Yeah, maybe. But if you want to talk to people who aren't doing marine stuff. But yeah, so it was really cool. Everyone was just super social, and it was just cool because everything was so interesting because it was something that's completely outside of what we do all day, every day.
Yeah.
I thought it was excellent.
Oh,
and
definitely.
then I also went to the bee tour. They had bees at Parliament House.
That does sound very exciting. What were your findings from this interaction in terms of like trying to get science to Parliament?
Bill Shorten was interviewed on the first day, and he was very frank and very funny. But some of the key points I got from his interview were to know your worth, like not just, I'm worthy. It's like know the actual dollar value of your work. So it's not just like, oh, we need corals because they're pretty. It's like, I know that, the Great Barrier Reef put in like $61 billion. I forgot.
Of tourism.
For like indirectly from, like all of those towns like Cairns and everything, it's tourism, but then all of the people who work off of other avenues of that as well. And then there's also the economic impact of like if we lose reefs, we lose all the fish that, need it for nurseries. So then it's like fisheries that go down. So just in WA, Ningaloo Reef, there's thousands of people who lose their job, if Ningaloo's gone, and it's millions of dollars for the WA economy if Ningaloo's gone. So it's huge.
Putting money values on environmental aspects is extremely hard and can be extremely variable. You miss one little factor, or you add or extrapolate one factor, it can like blow it out of proportion or not give it enough value. I can imagine that's a very hard thing to do. I know neither of us work in that space, but it's an interesting that he's said that.
Well, not like exactly, but
because something's money.
You can't just go to a policymaker and be like, oh, we need to conserve this. And they'll be like, why? And it's like, and it's like,
understand.
we can't put an oil and gas plant there, which is going to bring in billions of dollars for the economy because there's a reef there. And they're like, okay, that's nice, but like we need to make money.
Yeah, I know. That's it for me. Like that, if that was one of the main messages, is like know the value. Like it's sort of saying that all they're seeing is dollar. Like all.
Yeah, but it's also cultural
values.
Value as well.
Which you can't put money on.
No, but I mean, at least now.
And like, especially if we're still trying to learn these like flow-on effects from like one ecosystem to the other, and all that. Like, how do you put money values on that to say we need to preserve this? Like,
I think it's as well having that clear and concise picture of like, this is the impact. This is, this impacts this and this and this, big picture stuff, and make it really straightforward. So, for the whole build up to Science Meets Parliament, they were encouraging us to have a gaddy pitch of our work.
What's a
gaddy pitch? So, again, I don't have this off the top of my head, but it's kind of like that thing of, if I can remember it, my gaddy pitch for looking at turbid reefs is, how reefs all over the world are being impacted by climate change. Well, there are actually corals that are withstanding extreme conditions such as fluctuating temperatures and turbidity. My research looks at, looking at the adaptive physiology of these corals to help us better understand, how they can withstand these conditions. In fact, this could then be applied to future adaptations for other reefs to help sustain, so that's like the 4-part of this is a problem. There's a problem and it's like, well, this is what I'm doing about it. In fact, this is what it can like then go on to fixing in the big picture.
Right.
Really concise.
Yeah.
Like straight to the point, your key points, and it's like catchy. And
you definitely sold me on that.
Even though it was really
the start of it.
yeah, exactly. Because when you bring in that, like, like this thing's happening, and you're like, oh yeah, and it's like, well, this is what we're doing about it, and it's like, cool.
Yeah.
Instead of just being like, all the reefs are dying. Like sadness.
Yeah.
Give me money.
But they're not dying, are they? All of them? Some of them they're doing all right. Some
are doing all right, but like it's
overall, it's pretty pessimistic.
Yeah, and it's sad as well because any coral conference you go to, every single presentation starts with sea surface temperature anomalies and everything's bleached, and and it can be quite disheartening. Especially just before I came here, I was talking to a friend who's a coral researcher, and she's getting pretty sad about all of the bleaching in WA. And it's very heartbreaking, especially when they're, your local reefs and you love them so much, and seeing them in a state of distress is really sad.
Do you have advice to like ways to try and stay positive?
Yeah, well, I mean, you got to look at it from, at this stage it's just kind of like hoping for the best. I mean, it's hard though, because in WA what we're experiencing at the moment is so far more intense than the marine heat wave of 2010, and we saw impacts of that for years afterwards and all down the coast. And so it's kind of like we have this, it's not just this mystical, thing that's going to happen and be like, oh, we don't know like what these real impacts are going to be, because like we've seen it before, we've seen what's happened, and we know this is going to be worse. So it's just seeing at this stage, like how the full extent of how intense this is going to be, and taking the opportunity to learn from it as well and learn how the reef is either adapting or recovering, or hopefully what we can do to help it along and help sustain it.
This question might be a bit far-fetched, but do you think in our lifetime we're going to see the collapse of, like, say, coral reefs?
No, because I think we will see coral reefs won't disappear. They're not going to die. They're just going to change, So,
And whether they can change and adapt fast enough.
yeah, but it's the idea of this incredibly, dense acropora everywhere, super colourful reef. Like, people have got to. I don't think that is going to be as common.
Mm-hmm.
Reefs are going to be patchier. They're going to be covered in more of those stress tolerant genotypes. So they're going to look different, and they're going to be patchier, and they're going to be in conjunction with macroalgae and other stuff. So they're just going to be different. People have got to learn to appreciate, the
complexities, or what we've got, or both, I guess.
Yeah, both.
Yeah.
What the environment is able to hold on to with the giant boot that we have on its neck,
Be nice if we could take it off. I think.
Oh, wouldn't it just be so nice?
Yeah.
But I mean, it's that thing of we can't all of a sudden just.
No, it's the steps.
Yeah, but it's it's communicating it in a way that's understandable and digestible to everyone, because some people are very ignorant and very passionate about $1 today is worth more than $1 tomorrow. So why should we forego X, if we're not, sure what's going to happen next?
A lot of this show is based on the thought or motto or whatever's that you can't tell someone to change. They have to come up with the idea themselves. So this show is sort of based around to, like, educate, show, inspire, these kind of changes that are needed.
Yeah, and you got to kind of make it personal. That's another thing that came out of Science Meets Parliament is if you can make something relatable and make it like a personal connection with anything. So it's like, if, some climate denier, blah, 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 likes eating a certain kind of fish or something
lose that
for dinner. Yeah, well, actually that fish is going to not be around for much longer because it uses these reefs as a exactly. So it's finding that
humans.
Yeah. Finding the thing that makes it personal for all self-centered people to actually care about stuff.
Yeah. That's it. Yeah. Straight away I've had an example in my own head of something that I didn't think much about, but as soon as it started happening in my own backyard, I've started to be like, oh, no.
Yeah. 100%.
Yeah.
But I mean, it's unfortunate, but that's the way the human brain kind of works. You're just kind of, we're evolved and designed to take care of ourselves. So, it is hard to expect everyone to be, looking at the big picture of, what's going on everywhere, and for small island nations and, all of that stuff, when it doesn't have an immediate impact on you and you're not seeing it. It's that whole out of sight, out of mind thing. But, and then if you get so, I can get quite passionate about these things. And I think in the past, maybe this is something Homeward Bound has had with me as well, but in the past can get quite, irritated and angry and depressed about, why isn't why don't people care? Like, why is everyone just, I'm just going to live my life in my little house in suburbia and everything's fine. And why would I care about, this poor island nation that's about to go underwater and everyone's going to be climate refugees and blah, blah, blah. And I'm just like catastrophizing everything. Why doesn't everyone care? And that can be really just yelling at people and being, sad isn't an efficient way to actually help the cause, but then trying to, find those positive personal links and trying to communicate in a way that's uplifting and inspiring is so important. So, avenues, platforms like this is so important to try and
so you think this is important?
Spark that interest.
In the other science podcast platforms, yes.
Yeah, but yeah.
My two viewers, my mum and shout out mum.
Yeah. I don't know. I think it's that thing of trying to make everything's got to be like sexy for people to care about it.
Paramount importance.